In this video, I will be talking about aerobic metabolism and specifically how we get the acetyl-CoA needed to enter the Krebs cycle and the electron transport chain. So first, what is aerobic exercise? So the word aerobic means using oxygen. And specifically what this means is that the task that you are doing requires a certain amount of oxygen and your body is able to get to that requirement. No matter what substrate we start with, whether it's carbohydrate, fat, or protein, we are going to use the electron transport chain to make ATP from it. But before we're able to use the electron transport chain, we need to first make acetyl-CoA. So even though there are going to be the same steps of making ATP from carbs, fats, and proteins, the steps preceding the Krebs cycle and the electron transport chain are going to be a little bit different. The following slides are going to explain these different steps. So if we are starting with carbohydrates, either glycogen or blood glucose, we know that we first have to go through glycolysis, which is a 10-step process that takes glucose or glycogen and makes 2 pyruvates from it. There needs to be a step in between glycolysis and the Krebs cycle that makes acetyl-CoA from the pyruvate. This step is called the intermediate step. During this step, we are going to use the enzyme pyruvate dehydrogenase to make acetyl-CoA and NADH. So specifically, we are going to take the 2 pyruvates made at the end of step 10 of glycolysis and add 2 NAD pluses to it and yield 2 acetyl-CoAs and 2 NADHs. Those 2 acetyl-CoAs will move on to the Krebs cycle, and the 2 NADHs will move on to the electron transport chain. Next, let's talk about how we get acetyl-CoA from fat. Fat is going to be stored in a triglyceride, and the word triglyceride means a glycerol and 3 fatty acids. These triglycerides are going to be stored in adipose tissue, which is the fat that's just stored in your body, whether that's subcutaneous fat that's right underneath your skin or visceral fat that surrounds your organs. Triglycerides are found in lipoproteins, which is the scientific term for blood cholesterol, or directly in the muscle. The only part of this triglyceride that we're going to use in these steps are the 3 fatty acids. So the first thing that we're going to need to do if we're using fat as an energy source is to break the fatty acids from the glycerol. To do this, we are going to use the enzyme hormone-sensitive lipase to break it down. And this step is called mobilization. So during mobilization, we're going to take the triglyceride and we are going to use the enzyme hormone-sensitive lipase to cut off the 3 fatty acids from the glycerol. The glycerol left over is going to go to the liver, and the 3 fatty acids we're going to use in the next steps to make acetyl-CoA from it. This is going to happen outside of the cell. The 3 fatty acids are going to travel inside of the cell and are next going to have to get inside of the mitochondria, because in the mitochondria is where the Krebs cycle and electron transport chain takes place. To move inside the mitochondria, we are going to use the transporter protein carnitine. Carnitine it acts as a chaperone. It connects to those 3 fatty acids and brings it inside. And then finally, to get acetyl-CoA, we are going to go through a process called beta oxidation. And during beta oxidation, we are going to break off carbons 2 at a time. And each time we break down carbons and get 2 carbons from it, that 2 carbon chain becomes an acetyl-CoA. The amount of acetyl-CoA that we get from fat depends on how many carbons are in that fat molecule. So for example, most fatty acids are a 16 carbon chain. So if we have 16 carbons and we're breaking them off 2 at a time to make acetyl-CoA, that's going to give us 8 acetyl-CoA that'll move on to the electron transport chain. Finally, we can make acetyl-CoA from protein. We don't do this too often because protein has such an important role in our body. Proteins are used as transporters, they're part of enzymes, they're part of hormones, they're a major component of our muscles. So we don't want to use protein for ATP very often. However, in extreme conditions, so like if you're starving or if you're doing a high intensity exercise task that depletes all your other energy sources, you can also get ATP from protein. But again, in order to get ATP from protein, we first need to convert it to acetyl-CoA, which can go through the Krebs cycle and the electron transport chain. We're going to be able to break down protein using 2 processes. The first process is called deamination. And deamination means removing a nitrogen group. The second process that we could use is transamination. And transamination means transferring a nitrogen. By either removing a nitrogen or transferring a nitrogen, we will be able to make acetyl-CoA or glucose. If deamination and transamination results in acetyl-CoA, then that acetyl-CoA can move on to the Krebs cycle. If deamination or transamination yields glucose, that glucose is going to go through glycolysis and the intermediate step and eventually become acetyl-CoA. The process of making glucose from something other than glucose, so in this example, protein, is called gluconeogenesis. And this process is going to occur in the liver. Maybe some of you guys are wondering, how do I know if an amino acid ends up as acetyl-CoA or as glucose? It's just going to depend on the type of protein or type of amino acid. Some of them resemble fat when they get deaminated or transaminated, and those are the ones that go directly to acetyl-CoA, whereas some of them resemble glucose when they're deaminated and transaminated, and those are going to go through gluconeogenesis. So now, no matter if we started with carbohydrates, fats, or proteins, we have acetyl-CoA. And this acetyl-CoA will enter the Krebs cycle, which is explained in the next video.